Hello everyone and welcome to another MSD PigCast. My name is Stephan von Berg. I'm the Global Technical Director for piglet vaccines at MSD Animal Health, and with me today, and it's a great pleasure, is Jessica Barbosa. And so Jessica, please introduce yourself, tell the listeners who you are and who is Jessica.
Okay, thank you, Stephan, for the opportunity to be here. I'm very happy because it's my first time traveling far from the North and South America, so it's a very interesting place to be here. And my name is Jessica, and I'm from Brazil, a warm country. And I did my vet school there in Brazil, and I started my master in working with pigs. Actually, I started working with pigs when I was in my second semester in my undergrad, so it has been some years that I start in that field. And my family still ask me why I'm working with pigs. For them was a big surprise.
So why do you work with pigs?
That's a good question. I don't know what's happened in that. I remember I went to know the places and the swine farm at my university, and I completely fell in love with them. Actually, I had some contact, a previous contact with pigs in the farm when I was a kid, but just that. But thinking work with pigs was a really surprise for me because first I was thinking to work in a field in a warm arms from Brazil as a vet. So I changed a little bit my mind about it. And since that I entered in the research area there in Brazil, mainly work with nutrition as to control intestinal health. Mm-hmm. And I did my master focusing, looking for some additives and nutrition in controlling intestinal health. And I decided to move to another country, a cold country called Canada. And there I'm working with enteric disease. So I continue working controlling intestinal health, but now more focusing microbiome and enteric disease. So I have two professors that they support me, Professors Vinícius Cantarelli in Brazil and Professor Matheus Costa in Canada. So since then I started and I've never stopped more to work with pigs and intestinal health.
Perfect. And to give them some context, because Jessica mentioned the travel that she did, right now at the very time we are in the beautiful Valencia in Spain at an event. And so you moved into the pig area and intestinal health as your specialization and research focus. And it's a natural progression from pigs to nutrition because it's important and intestinal health. But for you, what is the point that sparks your interest in that exact topic?
Oh, good question. Well, for me, I'm always fascinated by physiology in general, but when I think intestinal health or intestine or gut in general, this is so important because this is we can call as our life machine that will help us to convert or to transform what the pigs are eating. So we will prepare everything there in nutrition. We have the intestine to help us or to work for us to transform what they eat in nutrients for them to grow, and in the end we have the pork and everything. So I think I'm fascinated by this, the intestine or the gut can do for us, transform the food and grow for performance or meat in the end. So I think this was what put my attention or catch my attention in swine production.
Yeah. Yeah, very, very, very relatable to that. And now, if you look at intestinal health, and you've been talking about the intestine as the growth machine, which is a picture that I really like because that's what it is. And we certainly see issues in our pigs, in our domestic pigs, in the farms with intestinal health. So from your perspective, what would you say, what are maybe the three most important problems that we see related to intestinal health?
Good question. So I think for us the three most challenges based on my opinion, what we are doing now, I think we have a good problem with, mainly in the wean phase with E. coli or the post-wean diarrhea. This is a big problem or a challenge for us to control, as we have a lot of factors involved there in that moment. We are having a big problem. We still have a big problem. I was thinking more in bacterial disease. Let's think about Lawsonia, for example, and swine dysentery caused by Brachyspira. But of course, we have other problems. But for me, I think this is it's a problem that we have around the world. And I think I can add more one, but we don't have, for example, in Brazil, we are lucky, but we have PED in Canada, United States, and other countries. So those are big challenges when we think in control intestinal health and the profitability of the farmers, in my opinion.
Yeah, I agree. And you mentioned weaning, and weaning is one of the most crucial parts of a pig's life, right? Because at that time, so many things happen, the stress, and it has an impact on the gut. We see that. We see a degradation of the microvilli, for example. So really an important point, and I think even looking globally, this is one of the most important topics that we need to address. And I know that your research group in Canada, you are looking into ways to address these issues. Can you give me examples of the methods or concepts that you are looking into?
Oh, yes. So in Canada, basically, my research line, my research group, we are trying to understand what's going on when we have swine dysentery, when the pigs are challenged or infected by Brachyspira dysentery or Hampsoni. And we are trying to understand what is the power of the microbiome associated with this disease, because we have some data that show us that there's some kind of bacteria that can help the animals to develop or the disease, that can help the Brachyspira to reproduce and cause the clinical size in pigs. But we have another group of bacteria that will help the pigs to overcome this challenge. So now my focus there is try to understand why some animals, after they are challenged with Brachyspira, why some of them they develop the diarrhea, the hemorrhagic diarrhea 3-4 days after the challenge, but why some animals they will just develop this diarrhea some days after. That is not like this following this protocol. 3-4 days after they will have this diarrhea. So I'm trying to understand what is the association between microbiome and the time to develop the hemorrhagic diarrhea. But there is another point of research there that we are trying to understand how can we improve or develop some vaccines against Brachyspira to control swine dysentery. I cannot say a lot about this field because this is not my project, but it's I'm into this. And we are having some interesting trials that in the future, we can show the data for everyone. So we are having some good results about it. How can we develop some efficiency vaccines against swine dysentery that is a problem for us? And now we don't have effective vaccines to control this. So it's basically these two topics, but I'm more focused in the microbiome and the relationship between microbiome and Brachyspira, for example.
Yeah, so side note, efficacious vaccine against Brachyspira would be a huge step and a huge improvement. That's for sure. So I'm happy to hear that in academia there's work being done on it. And you mentioned the microbiome and the impact of the microbiome in a very controlled setting. You said you have these pigs and you do challenges with Brachyspira. And you see different outcomes based on the microbiome. And what makes the microbiome from one animal different to the other?
So
is it the composition? Is it the diversity or non-diversity? So this is the line of thought that I have.
yeah, this is a good question that I don't have the answer yet for
Okay,
you.
sorry. Good. Looking forward to the next time.
Yes, but basically we can see some change in the microbiome profile when we have the animals develop the disease or not. But nowadays I'm working in the data and the bioinformatics staffs to have these answers for you and for us. So luckily, I think soon we will have these results to discuss more about it, because it's interesting how the microbiome, this complex word, can influence the disease or can influence other points or other topics when we talk about swine or animal production in general.
Yeah, yeah, and that's interesting. So you, of course, and as a scientific or research project, it has a very sharp but narrow focus, right? So impact on Brachyspira. But I mean, we need to be honest, microbiome right now is a hot topic, and we've seen that the amount of publications alone has an exponential growth, right? So as one of the researchers working with that, what is your thought or your, maybe let's call it educated guess on what the impact of understanding the microbiome better will be on the future of the swine industry?
Yes. I'm not an expert in microbiome, but I'm trying to enter in this topic, because I think, my opinion, this start to be and will be our future to control a lot of, to overcome a lot of challenges. But the things that I know is that microbiome it's so complex. We still don't know what is the best microbiome, for example, because we are looking for this. What could be the best microbiome for us to work with this and to play with this to help us? Or even we have a lot of environmental factors that will impact in this microbiome, especially in the beginning of the life of the pigs, that this phase that we can modulate or shape this microbiome, even with nutrition or some from the soils or from the environment. So we have a lot of factors that influence this microbiome. It's so complex. But I think the most important now is in not try to understand who is there or what the name of the bacteria is there, but what they are doing there. So I think for us it's more important to know than who is there, but what they are doing. I think when we understand more what they are doing, this will help us to to have more solutions or to play more with this complex environment.
Yeah, I fully agree, because what we know, for example, is that you may have the same composition of bacteria, but then the very same bacteria have a different gene transcription and they produce different proteins, which leads to a totally different environment around them that you do not capture when you just look at which bacteria are there. So I fully agree there, and that's it's a whole new universe to look into, to be honest.
Yes.
Related to this, do you have experience maybe from literature, not necessarily from own research, from literature or from other colleagues with, for example, if we have identified a non-sufficient microbiome in a piglet or a pig herd, would there be a method to improve that already existing, maybe also from human medicine or other related areas, let's say?
Oh yes, I think now the researchers start mainly thinking in human medicine. I think they are more advanced on this microbiome field and try to help them. So we have this fecal microbiome transplantation, or FMT. They have been doing this for humans.
That sounds a bit nasty, by the way, but
yes, like, the first time that I heard this, it was like, okay, how will you do it? It sounds a little weird. But they have been doing this or they have been exploring this FMT for a long time in human medicine. And as they have some good markers that they can control this, they can notice is a good or not microbiome for humans. We don't have this already for pigs, for example. So the FMT, basically we will select good donors. What should be these good donors? The people will pass for a screen to know if they have some disease, because they need to provide the best microbiome possible. So basically the feces from this health or select donor will be source of good or beneficial bacteria that will restore or work to improve the microbiome on environment for people that are suffering for some enteric disease, for example. And they have this very well documented, or they have this standard to use to control Clostridium difficile for humans, for example. And why not try to use FMT for pigs? But how we will do it? So the researchers, they start to looking toward this. There's some studies that they did this FMT from sows, for example, to pigs. So using colonoscopy, but now there is some recent studies that were try to do this orally by oral method. So they select this feces from the health donor or the health sows, for example, and they will these feces will pass for some steps. So they will do a filtration, and after we can store this, because before...
It needs to be safe as well.
It needs to be safe, this is the point. This needs to be safe because we are dealing with live microorganisms. So, and after we can give this for the pigs, and this health microbiome or FMT will help us to restore or to build a good microbiome for pigs, and this can help us to the intestine or the environment work properly. And I'm talking about sows because this phase of life for the pigs is important, that we can modulate this very easily. So why not try to do this in the beginning? But we have a lot of questions about it. We still don't have a lot of answers. Which pig will be our health donor? How can we select this, as we don't know what is a good or the best microbiome for us? Which method will be viable for us to use in a commercial farms? So it's not easy, or how can I say? We will not do colonoscopy in pigs, in farms. It's impossible. There's no way for this to work. Yeah. We can have this oral method gavage that we can do, give them by orally to help them. But okay, how can we improve this? So we have more questions than answers, but for sure this is a field. And another point, how many doses should we use to have a good answers or good results in the end? So there is a recent study that they tried to do just one dosage, and they saw a very interesting shift in the microbiome. But how long this will be working for pigs? Should we repeat this again in the future or not? So we have much more questions than answers. I think there is more expert people to talk about it, but for sure this should be or could be our future in swine, thinking in microbiome complex world.
Yeah. And right now we are, let's say, at the beginning of the microbiota area, right? Or not area, era. And now we are thinking about it in a way of fixing a health problem, right? So yes. that's what we are, what the focus is right now. Your work with Brachyspira and impact of the microbiome, and the transplant in humans with certain diseases. But could you think of a even further application of that in the pig production for let's say, manipulating the microbiome?
Yeah, there's a very interesting and good point. I think this is our goal. Okay, we talk about intestinal health, microbiome, and to control disease. But why this matter for us is because we need that our animals perform well until the end. So I think there's some groups, research groups, they are focused on specifically this, how we can modulate the microbiome or the microbiota to help us to improve performance or feed efficiency. And I'm not sure exactly what kind of microbiota we have to improve the feed efficiency, but I know we have some specific bacteria that we are having good results to improve feed efficiency, as Ruminococcus, for example. So in the end, all these microbiota, they will try to, the nutrients that the pigs are not, they are not transfer, the ingredients or the food that the pigs are eating, that they are not transform everything in nutrients. This kind of bacteria, they can work in this for take these nutrients for her own energy or source of to survive. And then we can, they can change the environment to improving our immune system, to keep our immune system not too much activated, because we have this beneficial bacteria working there. And these nutrients from this bacteria can help us to improve the intestinal barrier function. I think will be a complement. So this bacteria, this good bacteria that we are having, find to improve the feed efficiency, they will work to try to keep in this intestinal environment in the most health way possible. So I think in the end, this will be why we have this improving performance and feed efficiency. But I think this is the most interesting point when you think about microbiome, how we can improve performance. And yes, we can modulate this, but we're still walking and thinking and
early
focus,
steps,
yes, to looking what will be the best microbiome or markers that we can find and work with this to improve the performance. Yeah,
it's very interesting, and as I said before, it's a whole new universe. And then, if you think the host, the animal, if you put that into the equation. And you mentioned the immune system. I need to be careful because that's my area of interest and expertise. So, but what you need at regulation for the immune system to tolerate the microbiota but still attack pathogens, etc. That's a whole different world. And just quickly before we wrap up, we were talking now about the microbiome, the manipulation potentially of the microbiome, but we still have to deal with disease. And just quickly, because you also have been looking into human medicine and the advances that have been done there, are there any other methods that you could see that are happening in other fields that may end up being applicable in swine in the future?
Oh yes, sure. We have a very interesting approach called phage therapy or bacteriophage therapy, and basically this phage, they will kill the bacteria as they need a host, a bacterial host for them to survive. So in that case, they have the good point about the phage therapy is that they need the bacteria need to have this complementary receptor, so they will be specific for some, they will target specific bacteria in that case. And the phage therapy can help us, for example, to control a specific disease. There's a lot of studies focused on salmonella and control salmonellosis in humans or in pigs. In poultry, we have a lot of good results about it. So the phage therapy, basically, they will take advantage of this infectious cycle of this lytic phage. And in the end, that they will multiply in that bacterial host, and they will, in the end, they will be resulting in lesions or death of this bacteria. And this, of course, this is expensive for human medicine. They start to use this to control some specific diseases. It's expensive. I'm not sure, maybe there's some other people that know if we are start to use this in pig production, because we started to look for this some years ago. But I think, of course, this is too expensive. We need to understand more about it. What will be the amount of phage therapy? The best method, delivery method for pigs will be via water or feed or another method. So I don't know exactly about it. But I think for us will be a very interesting solution when we are dealing with specific disease or infections that we have multi-drug antibiotic resistant, for example.
Yeah.
So in that case, we don't need, or we will decrease the use of antibiotics, thinking about specific infections. So the phage therapy could be a very interesting, it's an interesting approach, but we can explore more them to keep this in a viable way for using swine production.
Yeah, yeah, of course. I mean, everything at the beginning is complicated and expensive, and with more experience and more technical advantage,
yes,
things become more affordable. If you look at sequencing techniques, I remember 10 years ago, 15 years ago, it was like you had to get an own funding to do it, and now it's just you can look at the whole microbiota for it. Yeah. And for the phages, I think that's a really interesting approach, and I like the thought of a self-replicating therapeutic, right? So that's also something interesting. So we are coming to the end, Jessica. It was really a pleasure to talk with you, and I'm sure we will have the opportunity to repeat that talk in the future with more insights, more results. Also from your own work, really looking forward to that. And thank you for being here.
It's a pleasure for me to be here. Thank you for the opportunity to talk about it. I really enjoy and talk about intestinal health and everything that we can do to control this or to make this working properly. And yes, I will be glad to talk about our new results or what is going on in other fields or in other research about intestinal health. And thank you very much for the opportunity to be here talking to you.
Great, appreciate it. Very well. Thank you for listening to that interesting episode, and looking forward to the next PigCast by MSD Animal Health.